Okay, welcome back. Last time we talked about protein structures. If you remember, we had primary, secondary, tertiary, quaternary structures. This is all different ways of describing the same shape. The primary structure was the amino acid sequence. The secondary structure was the ways in which those primary sequences, in which consecutive amino acids interact with each other in order to make a beta sheet or a alpha helix, whereas the tertiary structure was a mapping of all those secondary structures to map out an entire protein. And a quaternary structure is mapping out how individual proteins interact. So therefore, it's definitionally a multiple protein structure. Today we're going to move on to how proteins work. So a protein's function is defined by its shape. The shape of something determines what its function will be. Just the same way as you could try and put on lipstick with a mascara brush, but it wouldn't work so great. Or you can try and screw something in with a hammer, and it probably won't go so well. At best, an incorrect shape will result in a non-functional protein. So proteins that have the wrong shape can be involved with that's prion disease. That's what mad cow disease is actually just a protein that's got the wrong shape and somehow it managed to misfold other proteins like it. So the best case scenario for a protein with the wrong shape is just that it doesn't work. Some of the symptoms of Alzheimer's, we're not quite sure, or last I checked, we're not quite sure if it is a cause or a symptom of Alzheimer's, but these plaques, these beta plaques, they are just big groups of big clumps of misfolded proteins. So the shape of a protein is incredibly important. The thing that holds a protein together, the thing that keeps all of these different shapes together, these are all hydrogen bonds. Remember, we had our hydrogen. Let's say if we're making a helix, we could have this hydrogen. We could sort of twist this whole shape such that this hydrogen is above this oxygen, this hydrogen is above this oxygen. We have hydrogen bonds connecting them. So the thing that holds all of the protein shape together is hydrogen bonds, and hydrogen bonding is incredibly important here. Disrupting this, or removing the protein's natural shape, is called denaturing or denaturation. This is unfolding the protein or disrupting its three-dimensional structure through breaking its hydrogen bonds. And we can do this through temperature, obviously the same way that like boiling water worked, but it can also happen through pH or through radiation. And of course, destroying the protein shape will destroy its function. Mutations in the DNA can often result in mutations to the overall protein structure. Our classic example here is always hemoglobin. So this this tetramer, this four protein structure, or this four protein complex is called hemoglobin. It's the thing that binds oxygen inside your red blood cells, but it is made up of these four individual beta globin chains. Now, as the blood moves through your lungs, hemoglobin binds to oxygen inside your red blood cells, and it then delivers oxygen to other cells. After the oxygen is released, the red blood cells return to the lungs to get more oxygen. So that's what happens when we breathe. And the oxygen binding properties of hemoglobin exist because of its structure. So if we look at this here, these little red things, these are the heme groups. These are the actual things that bind the oxygen. The little things that are attached to, they sort of hang out. They're not protein bits, they're actually connected to the protein bit, but in any case, the thing that, the binding pocket for oxygen exists at the interface between these two proteins, and this one is between these two proteins, this one between these two. So you can't actually have the binding you can't bind oxygen unless these four monomers, or these four, not monomers, but these four proteins are come together in a tertiary structure. So the oxygen binding properties of hemoglobin exist only because of its structure, and specifically its quaternary structure. So although definitionally below that is each of the tertiary structures, each of the individual beta globin chains has to fold properly themselves such that they can all fit together like a very complicated jigsaw puzzle. Yeah, so each forms a pocket that holds an iron-containing heme group. So glutamate, we don't need to know specifics about the amino acids here, but there's one called glutamate. Glutamate has a negative charge, and valine, we can actually see here, is that we can see glutamate here has this carboxylic acid on the end. All right, we have this carboxylic acid here, and valine is neutral. It has no charge to it because we can see it's all CH, it's all methyl, it's all carbons and hydrogens. So that's all very nonpolar. Valine is a nonpolar amino acid, whereas glutamate is a polar amino acid. It's a charge. It has a charge to it. So this change from charged to uncharged, polar to nonpolar, actually causes this is a very extreme case. This is this rarely anything like this extreme happens, but this one amino acid change can actually changes the shape of the whole protein, and that's actually not uncommon. But the thing that is uncommon is that this amino acid change actually causes a shape in the entire blood cell. So usually blood cells have this little like button shape. They have this shape, by the way, because they don't have a nucleus anymore. This little space in the middle, that is where the nucleus was taken out, is in a process called enucleation, enucleation. And you end up with something that looks like this. So this hemoglobin mutation induces a hydrophilic to hydrophobic change, right? So we have this sixth position here. So hydrophilic to hydrophobic change, it actually changes it does, of course, change the form and function of the entire molecule, but it even changes the shape of the overall blood cell itself. And this is called sickle cell anemia. So red blood cells that have these abnormal hemoglobin molecules become distorted into a sickle shape. Some people might try and tell you this is like a black person disease or something. That's nonsense. This is in response to malaria. The selective pressure that sort of promotes sickle cell is that if you are heterozygous, if you have one mutated copy and one unmutated copy of the sickle cell gene, you have a resistance to malaria because they are codominant. But they are codominant alleles. But this isn't like specific to black people or anything. Like it's some major nonsense. So this is actually just exclusive to wherever people with malaria, or anyone who lives in a place with malaria, which includes sort of Eastern Africa all the way out to, or sort of north and mostly Eastern Africa all the way out to like India. But is not present. We do not see sickle cell anemia in places such as Kenya, which is in a very high altitude. Which is a high altitude country, so in which malaria does not survive. So it is not exclusive to like any skin color or any nonsense like that, right? So this is sickle cell anemia. Yeah. So that's it for proteins. Our next thing that we're going to talk about is nucleic acids, our final biological polymer or biological macromolecule. Are nucleic acids. These are also very important. They're all very important. But nucleic acids, these are built from nucleotides. The monomer in this is nucleotides. The language in which this is nucleic acids are the language in which all information about an organism is coded, or at least almost all information about the way that it looks. Nurture is in there. This whole nature nurture thing. DNA is not the be all and end all the way that some people like to think it is. There's a lot of other things that work, some of which are also nature, some of which are nurture. There's epigenetics, which are just sort of patterns of gene expression, and also energy molecules. If you remember anything called ATP from your biology classes in high school or whatever, this is the energy molecule, adenosine triphosphate. It is also a nucleotide, or is very similar to a nucleotide. So what about RNA? You may be thinking, right? So you've heard of DNA. You might remember RNA as well. So DNA is a genetic blueprint. However, proteins are specifically coded by RNA. We have this sequence of this progression of information within the cell. DNA is used to create RNA. DNA is not turned into RNA. It's just RNA is a separate thing that is created based on DNA. And then proteins are actually made by reading the RNA. This is because DNA is very important. Protecting the DNA, protecting the genetic code of an organism is like one of its major responsibilities. So DNA ends up being very stable. You can put some DNA in a test tube, come back in like 100 years, and it's absolutely fine. RNA will degrade just on its own pretty easily, even in the best conditions. So nucleotide, the basic structure of a nucleotide is made up of a base, a nitrogenous base, a sugar, and a phosphate group. I don't know why the S in sugar or B in base is capitalized. They're not proper nouns. Bases can have a single or double ring structure. So here you can see there are purines and pyrimidines. This double ring structure is called a purine. If you ever wondered why we have bases inside of nucleic acids, it's because they're named nucleic acid because they're an acid that was identified from the nucleus. Because again, scientists are very boring at names. But it is a nitrogenous base because the base of this compound is actually nitrogen. It's mostly nitrogen. So when they're discovered, they're like, oh, this is some sort of nitrogen-based thing. So it's just called a nitrogenous base. So there's no like chemical base inside of a chemical acid. It is a bunch of nitrogens inside of an acid. Pyrimidines. These are single ring structures, 6-ring structures. They are slightly different. DNA uses A, G, adenine, guanine, thymine, and cytosine. RNA uses adenine, guanine, cytosine, and uracil. Inside of DNA and RNA, they both have the backbone, the sugar phosphate backbone of DNA or RNA. The only difference is that we have an OH here versus an H here. Having this, so when more nucleotides are strung together, this OH here is actually used to connect new. This OH is used to connect the next nucleotide to. So what happens is that this the phosphate of the next nucleic acid is actually taken and it's added onto. Where's my mouse going? It's added onto here. I don't know if you can hear my dog barking upstairs. If you can, I'm sorry. So, hold on. What is going on? Okay, sorry, I'm back. There was the UPS delivery person was outside, and Zoe was not having any of it. He is not allowed around here. But he's gone now. Presumably, Zoe thinks that he she scared them off. So yeah, bases can have these single double ring structures. What was I saying? Yeah, so the phosphate from the next nucleic acid is added on here, such that we have this polymerization through here. And this OH is loose in ribose, but is only a hydrogen in deoxyribose. This is the only chemical difference between DNA and RNA, and this is because this OH is here, it's open for like things to come in and do chemical reactions with it, because we know OHs are a place where reactions are likely to happen. So this is where this is why DNA is so much more stable than RNA, is because it doesn't have that OH group there for something else to come in and like grab onto and do a chemical reaction with. So yeah, DNA stands for deoxyribonucleic acid. It's deoxy because it has had this oxygen removed. It's located in the nucleus, therefore it's a nucleic acid. It contains instructions for everything, just about everything a cell is and does, and it has a double-stranded helical structure. You can see, if you imagine to sort of take your hand and curl it around with your thumb facing up, your right hand will go along here with this, will go with this curve, whereas here it's going to actually cross over. Right? If your hand is here, it's going to cross over this helix. So DNA is always right-handed. DNA is never left-handed. These are the 4 nucleotides. Process of going from DNA to protein. DNA is taken and it is transcribed into RNA, and then it's translated into proteins. So this DNA to RNA to proteins, this is called the central dogma of biology. This describes the flow of information within the cell. but it can sort of give us a incomplete view of how complicated it is, because it's not just these things happen. That's not the be all and end all. But it's the actual sequence of the bases in DNA that encodes all the heritable information. This is because all of this, this sugar and phosphate, these are identical between every single nucleotide. The only thing that changes is the nitrogenous base. Adenine always pairs with thymine. Guanine always pairs with cytosine. If you have trouble remembering this, the easiest way to remember it is that the ones with the flat edges go together. Holy crap, what was that? I don't know why my mouse keeps doing that. And guanine and cytosine go together. Because they're the ones with the curly bits. It's sort of like a recipe, right? So if you have a sort of family heirloom cookbook and you don't want to bring it over to your friend's house because they're real messy and they're going to get stuff everywhere, what you would do is you would just make a photocopy of grandma's cookbook. And then you can make a copy of it, whether or not that's a photocopy, taking a picture of it, rewriting it. All of that is just taking the same information. So when we say DNA is transcribed, we mean it's the same information but in a new place. So it's DNA and RNA are effectively the same language. They're just slightly different in that instead of thymines, there are uracils. But then in order to translate it, remember translation means to go from one language to another. Translation is going from the 4-letter language of DNA into the 20-letter language of proteins. That's something completely different. So cells need proteins to grow, maintain themselves, reproduce, do all this sort of stuff. And that's it. That's it for proteins. Here's a recap. We have carbohydrates, lipids, proteins, nucleic acids. Here's a little cheat sheet for you. Subunits. This is what the subunit is and what the polymer is. And I will put this up on to Canvas. And there's no quiz today. I would prefer you all just keep working on those papers. I think that's a much better use of your time. I think that would be also just more interesting to you. I don't want to give you all a lot to do. You've got a lot of shit going on in your life. So stay safe. Have a good one. I will see you next time.